In ancient Egypt, small white dogs were placed in the beds of the sick, believed to possess miraculous healing powers. By 500 BC, the Greeks had etched their distinct spitz-like figures onto clay amphoras. This is the Maltese. For centuries, they were hoarded by European aristocracy. Monarchs like Queen Elizabeth I and Queen Victoria protected these pure white companions, turning them into living symbols of status and royal favor. To maintain that tiny, prestigious aesthetic, breeders narrowed the gene pool for generations. They intentionally shrank the animal's frame, creating a severe genetic bottleneck that would eventually change more than just the dog's size. Standing only 10 inches high, a Maltese perceives sudden movements and approaching animals as immediate threats. What looks like a behavioral quirk is often a physiological attempt to deter perceived predators and artificially expand their safety margins. Centuries of selection for this specific size and coat came with an invisible trade-off. Deep within the breed's biology, the same genes that make them small are physically tied to a complex set of neurological, vascular, and cardiac failures. The origin of this biological trade-off sits on canine chromosome 15. Breeders focused intensely on the IGF-1 gene. This is the primary biological determinant for diminutive size. By selecting for this gene, they successfully produced the toy phenotype, but the process, known as a selective sweep, has a side effect. By forcing the size gene into the entire population, breeders accidentally dragged physically linked mutations along with it, locking them into the breed's genetic makeup. One of those hitchhikers is a mutation on the FBXW7 gene. This corrupted immune regulator causes the dog's own immune system to bypass normal checkpoints and attack its brain tissue, leading to a fatal condition called necrotizing meningoencephalitis. In the Maltese, the genetic desire for a toy-sized dog created a package deal that included a high risk of neurological ruin. That same genetic sweep is tied to vascular anomalies that form while the puppy is still in the womb. This is an extrahepatic portosystemic shunt. In a healthy dog, blood from the intestines goes to the liver for filtration. In a Maltese with this shunt, that nutrient and toxin-rich blood bypasses the liver entirely and dumps directly into the systemic circulation. The fallout is severe. Unfiltered blood floods the brain with toxic ammonia, causing mental dullness and seizures. Simultaneously, the body struggles to maintain glucose, triggering acute hypoglycemic crashes. To maintain its exterior aesthetic, the internal anatomy was structurally compromised, leaving many dogs in a state of chronic toxic poisoning. Centuries of artificial selection for human proximity left the Maltese entirely unable to maintain emotional equilibrium when alone. When separated from their owners, these dogs enter a state of acute biological stress. This triggers a massive chronic release of catecholamines, a constant state of internal panic. This data visualization compares a healthy heart to one under chronic separation stress. The constant fight or flight response leads to measurable remodeling of the heart muscle, including hypercontractility and ventricular dilation. Resolving these panic responses is a medical requirement to protect the heart from progressive damage. All of this creates a complex diagnostic labyrinth. When a Maltese presents with a seizure, a clinician must determine if it is primary brain necrosis, toxic liver blood, a metabolic crash, or a physiological result of extreme panic. This is complicated by human size biases. Veterinarians often assume large dogs are tough and under-medicate them, while the genuine physical pain of a Maltese is sometimes misinterpreted as behavioral acting out. This chart plots quantitative sensory testing, which measures exactly when a dog's nervous system responds to stimuli. The results show that pain thresholds are not dictated by size or perceived toughness. The Maltese possesses measurable physiological pain limits that function irrespective of human bias. Effective care requires looking past basic training. Because their panic is rooted in neurochemical depletion, these dogs often need targeted pharmacotherapy, like fluoxetine, to lower the biological threshold for panic. True care for the Maltese requires moving past the purse dog image. We have to respect the severe, complex biological reality we engineered into them.